Good afternoon, everyone. Welcome to today's webinar held by ALS Malaysia on microbial source tracking, identification of microbial contamination in water body. My name is Nujin. I'm your speaker for today. If there is any questions, I appreciate you to ask me at the end of my presentation by typing in the questions. So before I move on to my presentation, here is a brief introduction about ALS.
Welcome to ALS Malaysia's presentation. This is a photo of our building, which is also ALS Malaysia's headquarters in Bukit Jelutong Shah Alam, Selangor. Before we start, I would like to show this slide as a reminder of safety in everything you do. We uphold and practice a culture of safety, as safety is one of our ALS core values. In this time of COVID-19 difficulties, please practice safety first culture in everything you do. ALS stands for Australian Laboratory Services, and its history started back in 1863, originated from Brisbane, Australia. We have presence in more than 65 countries with 400 plus laboratories worldwide, employing more than 13,000 people. In Malaysia, the management is headed by our General Manager, Dr. Chin Tin Tin, with a total staff strength of about 200 people. We have a total of 4 laboratories in Malaysia, with the HQ in Bukit Jelutong, Shah Alam, Selangor. Up north we have a lab in Penang, down south in Johor Bahru, and East Malaysia in Kota Kinabalu. All our laboratories are also MSISO 17025 accredited for food chemistry, microbiology and environmental testing. We also have 3 sites offices at Kemaman, Ipoh, and Bintulu. ALS Malaysia has garnered several recognitions over the years as a certified MSISO 17025 laboratory. We are the recognized panel laboratory for Ministry of Health and Food Safety Testing, National Pharmaceutical Regulatory Agency, Jacob's Halal Panel Laboratory, Ministry of Environment and Water. Department of Fisheries, Certified GMP Plus Laboratory for Heavy Metal, Mycotoxins and Dioxins Testing. We are also the BioNexus Partner Laboratory for Bioeconomy Malaysia, providing testing services for all the BioNexus companies in Malaysia. ALS Malaysia was also awarded the IKM President Laboratory Award year 2017 to 2020 in the category of Best Commercial Testing Lab. These are some of the certificates mentioned from the previous slide. Thank you. Welcome to ALS.
So in today's presentation, I will give an overview of microbial source tracking, the potential applications of microbial source tracking, some case studies, as well as the tiered approach testing methodology. So this newsletter was published sometime in the middle of last month. So the studies was conducted from November 2018 until last month on the groundwater in southwestern Wisconsin. And what they found out was that 105 out of 138 well samples had signs of contamination, whether that stemmed from humans, cattle, or pigs, that exceeded the state's health standard. So a higher percentage had human-related pathogens, with 46% of the wells showing contamination from human fecal matters, 24% of the wells showing contaminations from cattle matter, and 9% of wells from pig fecal matter. So another almost similar findings where fecal contamination happens in beach rather than wells. So this event happens sometimes around end of August at one of the beach in Portals Lake, where the beach was closed due to a high bacterial levels in the water due to fecal contamination and high temperatures. Back in home, we are also facing the same problem. Last August, water discharged into Sungai Kreh were black in color, and they have this very unpleasant smell, which was believed to be from swine manure from the Kampung Selamat pig farms. So this is the picture on how bad the pollution is in Sungai Kreh. So another local headlines that happened last Sunday at Sungai Semenyih and Sungai Langat, which lead to water supply disruption in several areas. It was suspected that the source of other pollutions in these two rivers are due to duck and goose farms. So this is what our water quality specialist, Dr. Zaki Zainuddin, had to say about the pollution that happened last 2 days.
Sungai Batang Benar dan Sungai Pajam sememangnya terdedah dengan risiko pencemaran daripada pelbagai punca bahan cemar. Pakar kualiti air dan permodelan Insinyur Dr. Zaki Zainuddin berkata, ia berikutan dua sungai itu terletak di lokasi industri, penternakan dan tapak pelupusan sampah.
Menurut Dr. Zaki, apabila ada punca bahan cemar berkenaan, risiko pencemaran tidak dapat dielakkan.
Well, untuk kes yang berlaku semalam sebenarnya, kalau kita tengok di hulu di Sungai Batang Benar dan di Sungai Pajam tu sebenarnya ada berbagai-bagai punca pencemaran di sana. Bukan saja pencemaran industri. Seingat saya berdasarkan kajian yang saya jalankan di sana, selain daripada punca industri, ada juga punca peternakan yang melibatkan peternakan ayam, peternakan daging. Ada juga kolam ikan di sana, ada juga tapak pelupusan sampah di sana, dan ada berbagai-bagai punca yang berada di bahagian hulu sungai. Jadi ini sebenarnya mendatangkan masalah bila kita.
So in today's webinar, I'm going to bring you through how can we apply microbial source tracking to track this contamination, and most importantly to solve and curb organic waste or fecal contamination in water. So organic waste such as feces represent a major source of water contamination because it may contain potential pathogens. This was also mentioned by US Environmental Protection Agency, where pathogen contamination by pathogenic bacteria and protozoan as well as viruses is a main issue for all types of environmental water bodies. Every year, millions of lives are lost worldwide, with a chunk in developing and underdeveloped countries as a result of waterborne diseases. Diarrhea is one of the major causes of death of more than 2 million people per year worldwide, mostly children under the age of 5. In fact, each day, nearly about 1,000 children die due to preventable water and sanitation-related diarrhea disease. And so, the next question will be, where do these fecal contamination come from? The source of fecal contamination in water bodies can enter streams from illegal or leaky sanitary sewer connections, poorly functioning septic systems, and poorly functioning wastewater treatment plant effluent. Besides, a significant amount of fecal contamination released in the waste is produced by animals. This can be a serious problem in water near cattle feedlots, farms, and dairies that have poor animal keeping practices, as well as waste if their waste is not properly contained. In urban area, fecal coliform can be contributed to surface water by dogs, cats, and human waste when it is carried into the storm drains and lakes during storms. Besides, runoff from vegetation's area, the stormwater runoff from urban, commercial, and industrial lands also discharges pathogens into water body. In addition, high amounts of sediments are often related to high concentrations of pathogenic bacteria. So the bacteria can attach to sediment particle. Fast-running water can also carry more sediment, so higher level of bacteria can occur during high runoff event. So bacteria are much more abundant on soil than water. Moreover, higher temperature and higher levels of nutrients can also increase the growth rate of bacteria. So why is it then important to know if the water around us is contaminated with fecal matter? This is because the presence of fecal coliform bacteria in aquatic environments indicates that the water has been contaminated with the fecal material of man or other animals. So at the time this occurred, the source water may have been contaminated by pathogens or disease-producing bacteria, protozoa, or viruses such as hepatitis A virus, Vibrio species, Salmonella, Campylobacter, protozoan parasites, which can also exist in fecal material. And so we assume for our own protection that the risk of transmission of pathogens may increase even though we may have no direct evidence of the presence of specific pathogen. Moreover, the risk of consuming pathogen from animals or human excrement is the most common and most severe of all threats to human health that come from contaminated drinking water. Fecal contamination can cause gastrointestinal infections following ingestions or infections of the upper respiratory tract, eyes, ears, nasal cavity, and skin. So in addition to health effects of polluted drinking or recreation water, there may be deep financial impacts as well. Economists have estimated that a typical swimming day in the Lake of Michigan Beach is worth approximately $37,000. So the economic loss for each day on which a beach is closed or under advisory for water quality problems can be quite significant. Worse still, fecal contamination water not only affect human health, but also marine and shellfish health. So finding from fish in Ethiopia, where pathogenic E. coli strain was detected in them, this may result in health problems. For those who love to consume raw and undercooked fish are more vulnerable. This will also lead to extra expenses like freezers, generators, the establishment of fish processing plants, and on-job training about proper fish handling practice to reduce the level of contamination of fish. Now that we know that fecal contamination of water bodies are a growing threat to human health, tourism, and the food industry, how do we then determine if there is fecal contamination in these water bodies in order to practice the best management to mitigate fecal contamination in them? So it is through traditional bacterial indicators, also known as FIB. FIB are pathogen indicators, as that term is defined by Clean Water Act as a substance that indicates the potential for human infectious diseases. FIB levels have long been served as a surrogate measure of fecal contamination and thus the presence of pathogens that are commonly associated with fecal material. So this method was recommended to be used back in 1970s by environmental protection agencies, where they will study the total coliforms level to fecal coliforms. So coliform bacteria are organisms that are present in the environment and in the feces of all warm-blooded animals and humans. Coliform bacteria will not likely cause illness. However, their presence in water bodies indicates that pathogen could be in the water system, as most pathogens that can contaminate water supplies come from feces of humans or animals. There are 3 different groups of coliform bacteria. Each has a different level of risk. So the total coliform bacteria are commonly found in the environment, such as soil and vegetations, and are generally harmless. Whereby the fecal coliform bacteria are a subgroup of total coliform bacteria. They appeared in great quantities in the intestines and feces of people and animals. So the presence of fecal coliform water bodies often indicate recent fecal contamination, meaning that there is a greater risk that pathogens are present than if only total coliform bacteria is detected. E. coli is a subgroup of E. coli is a subgroup of the fecal coliform group. So most E. coli bacteria are harmless and are found in great quantities in the intestines of people and warm-blooded animals. Some strain, however, can cause illnesses. So the presence of E. coli in water bodies almost always indicate recent fecal contamination, meaning there is a greater risk that pathogens are present. Most outbreaks has been caused by a specific strain of E. coli bacteria known as E. coli 0157. So this picture on the right shows the coliform bacteria colonies. So sometimes later, environmental protection agencies conducted a series of epidemiological studies to evaluate several additional organisms as possible indicators of fecal contamination, including E. coli and enterococci. So from the epidemiology studies conducted, it shows that E. coli is a good predictor of gastrointestinal illnesses in fresh water, and enterococci are good predictor of gastrointestinal illnesses in fresh and marine water due to their salt-resistant characteristics. And this traditional FIB is still being used until today. Although FIB are recommended for water monitoring because of their correlation with gastrointestinal illnesses, but they have their limitation too. It is difficult to differentiate sources of fecal pollution using these traditional FIB. Since coliforms and E. coli are present in both humans and animal feces, which means if we collect recreational water that consists of domestic pets, wildlife, and human, or any local water body that consists of urban agriculture or stormwater runoff, or even runoff from concentrated animal feeding operation or faulty septic system, retention treatment basin overflows. But what we will see from the traditional FIB is either E. coli, fecal coliform, or total coliform, depending on what type of broth that we are using. But you were not able to tell the source whether it's from domestic pets, wildlife, human, or agriculture. Another concern is that the fecal coliforms can survive outside hosts for long periods and even replicate after they are released into the receiving water, arising numerous heated controversies about the accuracy of these methods. Plus, the correlation between the presence of indicators and human health risks is also typically not strong. Moreover, knowing only the level of contamination without knowing the source of fecal contamination provides very little information to correctly assess and properly manage and remediate the human health risks associated with water contamination. And also to identify the origin of non-point source fecal contamination. So then the next question is, how do we determine the source of fecal contamination? It is through microbial source tracking, also known as MST methods. MST is a method designed to isolate, identify, and measure a host associated fecal indicator from an environmental sample. So the principle behind this method is that certain microbes, chemicals, and metabolites can be associated with specific hosts, assuming that human and animal communities utilize different substances. This might favor certain microbes in their guts than others. And so they will produce different byproducts that can subsequently be traced back to the source of the pollution in the environment. And that identified characteristics of these host-associated organism can be used as marker for fecal contamination from the host. And what motivates this type of study is that it determines the extent to which fecal sources influence human health risks from contact with water, as well as to attribute fecal loading in water bodies to the correct source, so that management and remediation efforts could be allocated in a more time and cost-effective manner. So MSD methods are commonly classified as library-dependent methods or library-independent methods. Both of these methods can be culture-dependent or culture-independent. And all these mentioned methods, you can apply genotypic methods, which I will explain further later. You can also apply phenotypic methods like antibiotic resistant profile or carbon utilizations profile, and also chemical methods like laundry detergents such as fluorescence whitening agents. So this is how a library and culture independent genotypic workflow looks like. This method is more sensitive, rapid, and easy to perform. So first, you will need to collect samples of water that you wish to test. Then extract the DNA and type fecal indicator bacteria from the feces of the animals judged most likely to contaminate the area, using host-specific markers and source identification, as in whether the water is contaminated by human, cow, or pig, as an example, in order to locate the water is contaminated by domestic wastewater treatment plant, dairy farming, pig farms. So why do we care about knowing the different sources? This is because so that we can accurately address the contamination issues and prioritize the site where water contamination occurs. MSD can also use to evaluate the best management practice to curb fecal contamination in water. It also supports the development and implementations of total maximum daily loads, where we can use this type of data to identify the maximum amount of a pollutant that a body of water can receive while still meeting water quality standards, thus lowering health risks associated with fecal pollution. On top of that, MSD also allows us to develop effective remediations and mitigation strategies and better assess human health risks associated with fecal contamination. Let's look at some successful findings that benefits from MSD findings. So this studies was published last 2 years from 2 major agriculture areas in Republic of Korea to track fecal sources in aquaculture areas. The southern part of Korea Peninsula is actually the major aquaculture area, producing more than 70% of domestic seafood for Republic of Korea. Hence, preventing fecal contamination of shellfish area is of particular importance because of the close link between water quality sustaining shellfish and the human food supply. This map here shows the location of which 240 water samples were collected over a period of years. So the author tested these water with 4 different host Bacteroides markers, namely the human, poultry, pigs, and ruminant. And what they found was the host-specific Bacteroides markers were detected widely in both the tested islands. So among which where the poultry-specific Bacteroides marker was detected at the highest concentrations. The mean concentrations and occurrence of the poultry and pig-specific bacteroides markers in the surface water of the island were higher than those in the bay, so which may have been affected by animals nearby. The sampling sites near residential areas also show high mean concentration of poultry and pig-specific bacteroides markers, which due to the characteristic of this area at which small-scale livestock breeding occurs in the residential areas, as well as the application of fertilizers containing human and animal feces. This is another study that published 3 days ago. This was conducted at the adverse aquifer that serves a primary source of drinking water to more than 2 million people in South Central Texas. So these studies aim to determine the primary sources of fecal pollution along Balcones and Leon Creek within the Edwards Aquifer recharge and contributing zones using general markers like E. coli, enterococci, and also universal bacteroideals. And also host-associated specific markers, human, dog, cow, and chicken and duck-associated bacteroides. They also based on the surrounding land use as a potential source predictor to help devise effective mitigation measure to protect water qualities. And what they found was the level of the 3 general indicators were highest and exhibited similar trends across all sampling sites. This suggests that solely by using these general markers is not sufficient for specific fecal source identification. So by using this specific marker, they found relative lower concentration for human-associated markers. However, the levels were higher in the developed Leon Creek area and highest following rainfall events. On top of that, relatively higher levels in human-associated markers were also observed at sites having greater human population and septic tank density, and may be attributed to leak or breaks of these infrastructures. Among the other host-associated marker, highest concentration were observed for dog markers in the Leon Creek area and cow markers in the Balcones Creek area due to farms. Chicken, duck, dogs, and cow markers also all exhibited higher concentration during the spring season and the end of fall or early winter, where markers levels exhibited stronger correlations with rainfall at sites with higher water resistance cover. So let's look at the testing method. So ALS can evaluate water bodies from the likelihood of different sources of contaminations. So what we do is we apply tiered approach, since this approach is most applicable when using MST methods. So this approach involves land use information along with some chemical tests, FIB and MST test. So first, you will need to identify the point source and then focus on the non-point source pollution that finally depositing them into the water bodies you wish to test. So what is point and non-point source? Point source pollution is any contaminant that enters the environment from an easily identified and confined place, like pipes, ditch, ship, and restaurant disposal. Whereas non-point source pollution is the opposite of point source pollution, with pollutants released in a wide area like excess fertilizers, herbicides, and insecticides from agriculture lands and residential areas, or oil, grease, and toxic chemicals from urban runoff and energy production, or bacteria and nutrients from livestock, pets waste, and faulty septic systems. So once you have identified all these point and non-point source, then you will then collect water samples from different sampling points. For this, you will have to decide. For example, you may collect from the downstream of the discharge point of domestic wastewater treatment plant, or at the juncture of a rivers, or downstream of a particular farm, and most downstream of all sampling points. So what we need is just about 1 liter of each sample from each sampling point, collect into a sterile bottles, deliver on ice to the lab within 24 hours. Once we receive the sample from you, we will then screen the water for E. coli and enterococci using FIB method. We will also check on the ammonia level, turbidity, and pH of the water. For E. coli culture, we will filter the water through filter membrane and roll the membrane onto the Chromocalx coliform agar. This is a chromogenic agar, meaning it is based on the color to differentiate E. coli and coliforms. So this is an example of it. So E. coli will form violet colonies and non-E. coli will form pink colonies. As for enterococci, we will roll the membrane onto KF agar. So this is an example of the agar. So if any spotted pink and red colonies are enterococci. So if these parameters exceeded a assigned threshold, we will then evaluate with more parameters, such as E. coli, enterococci, ammonia, turbidity, pH, bacteroideals, human bacteroides markers, and animal bacteroides markers. If you notice, we include these 3 more additional tests on top of all tests in the previous slides if some of the parameters mentioned in the previous slides exceed a assigned threshold. All these 3 parameters we analyze using PCR methods. So what is PCR? PCR is a method used to make millions to billions of copies of a host-specific DNA sample, allowing prompt and accurate identification of the origins of fecal water contamination. You might then ask, why do we choose Bacteroides? This is because this group of bacteria is most abundant in the human microbiota and also other warm-blooded animals. They are anaerobic physiology, prevents them from reproduce and survive in the environment. And most importantly, it allows species identification between animal hosts. So this is the workflow for how we determine the origin of fecal water contamination using MST. So first, we will collect sample, or you can provide us the sample, and then we concentrate the bacteroides through filtration through filter membrane, since bacteroides are very much diluted in water sample. We will then remove impurities and purify nucleic acid from the concentrated bacteroides material. Finally, we will target specific diagnostic sequences within the Bacteroides 16S RNA gene present in feces from different animals using qPCR machine. Currently, we do offer universal bacteroidals, and also for individual specific markers, we do offer humans and ruminants. But others like porcine, sheep, bird, horse, or any other animals that are of your interest, you can always contact us. We will then help you to design the specific marker for you and also run the test. So for those who wish to look at the entire microbiome community, you may opt for next generation sequencing. So some of the take-home messages for everyone. Organic waste such as feces contamination of water is commonly an enduring problem. Unfortunately, testing water for all possible pathogens is complex, time-consuming, and expensive. Hence, it is more reasonable to apply tiered approach testing methods combining land use information, chemical, and FIB method for screening, alongside with MST host-associated genetic markers if needed to find the source of contamination. Since MST methods pointed to specific contaminants sources that pollute specific water bodies and allowed more specific recommendations for improving water quality and safety. So I've come to the end of my presentations. Before moving to Q&A, here is other key testing services that ALS offer.
ALS Malaysia offers a wide range of analytical testing services, and these are our key testing services: environmental testing, industrial hygiene, food safety testing, biotechnology, medical devices and pharmaceuticals, and tribology. We thank you for your time, and we would like to take this opportunity to introduce our dedicated client service team that has been serving you all this while. Here's a short video from them, and we thank you again for your continual support. Thank you.
Thank you for using ALS services. We are your client service team.
Thanks for your attention. If you have any question, you may type in, and I'm happy to answer any questions. Okay, I have one question from Yong. How much sample needed? We need approximately 1 liter of water. So it will be good if you can provide us like 500 ml, and then another 2 is 250 ml separately in a sterile bottles. As for TAT, we take this as a project basis, so it's very much depending on your sample types and how easy or how difficult for us to handle the sample. So we don't have a specific TAT for this type of studies. I hope I answer your question. And I also got a question from Muhammad Azrul. First question is, what is the usual frequency and number of sample required for a meaningful and conclusive MST results in water bodies suspected to polluted by swine? Manuel, a lot of studies, they will actually collect every day in the morning, and they will do the study for about 1 year or 2 years just to really look at the whole pattern of the pollutions. So once you establish data for that particular water body, you can then use that as like a you can then use that as like a guideline for you, as in like how frequent then you have to do that. As for question number 2, how can we discriminate swine? How can we discriminate microbes that are related to swine manual than other animals like poultry, cattle, or even human? For us now, we're actually using Bacteroides markers. If you have noticed just now, I did mention that in different hosts, they will have different Bacteroides. So from there, we will be able to pinpoint. But as for like, let's say for human, we will sometimes use 2 markers just to confidently discriminate is actually from human versus to non-human. And also swine, poultry, cattle, we actually have these Bacteroides. On top of that, sometimes we will use mitochondrial DNA. So it's very much depending. Once you tell us what are the hosts of your interest, we will then look at which primer is the best to discriminate the host that you would like to discriminate. And how many specific markers are for swine down related microbes? So let's say once we design a primer for swine, we will test on different hosts. If it is very specific for swine, then we will then take that as a specific markers. So we don't say how many, but if that particular marker is not that specific, let's say it has cross reactivity with others hosts, we will then on top of one more, we will then use another combination of 2 primers in order to differentiate them. So it is very much depending. So and also the third question is what are specific species that correspond to swine manual in water body? As I mentioned just now, for Bacteroides, actually they have certain segment within the 16 RNA gene is able to differentiate among the hosts. So that from these 16 sRNA, from these 16 rRNA, we'll be able to know whether it belongs to other ruminant or not, because any other ruminant like cow, goat, or others, they actually have their specific ID within this 16 RNA gene. And question number 4, can MSD provide data that enable categorizations different sources and pollutant or microbe level? As long as it is from organic, we will be able to. But if it's like chemical, we will use some chemical testing to see what are they. But if it's organic matters, then yes, we will be able to note that it is from what type of is from which host. As for microbes level concentration or percentage, depending on what is your study objective. Let's say your study objective is you are going to collect from different sites and you want to see how many, let's say you collect from 10 sites and 7 sites that you found there are ruminants contaminant. So you will say 70% of the place that you have collected is belongs to ruminant. But if this is not your objective, for example, you want to know like how many copies are there, then we will need to do a more thorough study whereby we will need to study the gene copy number. Concurrently, we will do some bacterial plating to correlate what is the level of the colonies corresponds to the copy number. So for example, in the future, you would like to put like if ruminant has more than 100 colonies, is actually your cutoff point. Then we will see whether these 100 colonies of ruminant is actually equivalent to how many copy number in the qPCR. Then from there, next time you just have to run qPCR. So once you see that it reaches that particular cutoff point of the amplification graph, let's say for example at CT 35 is equivalent to 1.06 copies per mil, and that particular is actually equivalent to 100 colonies, then you can call that as your cutoff point. So it's very much depending on your on the objectives of your study. So the fifth question is where is the best sampling points in the case of rivers suspected to be polluted by swine manure? I would say that you will need to collect from the point where there is this swine farm, and then along the way until it reaches to the end of the river. So you need to collect from each of the sampling point. The more sample you collect between distance, you will see a better picture. When is the best time for sampling? Some people recommend in the morning, but we do also think in the morning. So rainy season sometimes will dilute the amount of bacteria off. But if it's for during rainy seasons, then we can just collect and we will concentrate it. So yeah. And also immediately pollutant is discharged or any specific time frame. We don't have a very specific time frame, but since we are looking at bacteroides, this bacteroides is actually anaerobic. So it can actually live 2 to 3 days in high temperature. But since we are looking at their nucleic acid, they actually persist longer in the water. So not too long from the date the pollutant is discharged. I would say maybe within 5 days. So the last question is regarding host-specific markers. Is it sensitive to various pig species or only to domestic pig? I would say that ours is more on a general pig. Yeah, we don't really discriminate between the species. So for like these type of studies, we will recommend you to let's say if you see any contaminations happen, immediately you collect the sample. That will be best, and then also to build a profile of the entire to build a profile for this area from upstream all the way to downstream. So in the future, once you see any pollution, immediately you just need to take one side and then you will then match back the profile. That will give you a better idea. We have this one question. What is the best management practice to reduce E. coli level in water bodies from Nurul Amira? So sorry that I don't have an answer for these questions as we only focus on testing. We don't really deal with this management practice. I really have no idea. Is there any more questions? So if there is no question, I'll end the webinar for today. Thanks everyone for your time. Thank you. Bye-bye.